So most organisms do cellular respiration in the presence of oxygen to make lots of ATP. But there are organisms that can survive without oxygen, either temporarily or indefinitely. So we're going to talk about what happens when there's no oxygen present. As we mentioned before, glycolysis directly doesn't need any oxygen. And so some organisms will do glycolysis and then continue to get a little bit of energy out of the remaining pyruvate through a process called fermentation. There's also anaerobic cellular respiration, which still does all the steps of cellular respiration, but instead of oxygen as the final electron acceptor, the final electron acceptor is some other molecule like sulfate. So sulfur-fixing bacteria can actually do this across their plasma membrane in a very similar fashion to our mitochondria. So we're going to talk in particular about fermentation, which happens in the cytoplasm and continues to produce ATP through substrate level phosphorylation. So if we think about what happens when oxygen is present, glucose goes through glycolysis to produce 2 pyruvates. And we make a little bit of ATP in this process, and we make our reduced electron carrier NADH. Then NADH brings the electrons to the electron transport chain and passes the electrons to oxygen. But when that happens, and it gives away its electrons, what we get back is more NAD+. And so as long as there's NAD+ present, then glycolysis can keep happening. But if there's no oxygen, then there's nowhere for NADH to give its electrons to, which means that we only have the reduced form of NADH, which means that glucose can't give away any more electrons, which means we can't even make that little bit of ATP that we get from substrate level phosphorylation. So the purpose of fermentation is to regenerate this NAD+, to strip the electrons out of NADH so that we can keep glycolysis going. And this looks different in different organisms. So in yeast, they do alcohol fermentation. In alcohol fermentation, ethanol is made as a side product. In our muscle cells, we do lactic acid fermentation, where pyruvate is converted to lactate. So this picture looks really busy here, but the big thing that I want you to concentrate on is, 1, glycolysis happens the same, whether it's alcohol fermentation or lactic acid fermentation. Glycolysis is still happening and making pyruvate, which means that NAD+ is receiving electrons and forming NADH. Second point, the purpose of fermentation is to regenerate NAD+, to convert NADH back to NAD+. And so how this looks is a little bit different in alcohol fermentation versus lactic acid fermentation. In alcohol fermentation, the pyruvate actually gets partially broken down and carbon dioxide is released. And so that carbon dioxide produces the carbonation that makes the bubbles that make your bread rise when you're baking bread with yeast, or that make the carbonation in your beer. It's because of yeast doing fermentation. Now, the yeast that are in our bread and our beer are then going to convert that 2-carbon acetaldehyde into ethanol. And it's actually this step where the acetaldehyde receives electrons from NADH that produces ethanol and reproduces NAD+. So, for alcohol fermentation, glycolysis happens like normal. We make NADH like normal, we make pyruvate like normal, but the additional steps are alcohol fermentation produces carbonation, which makes our beer have carbonation and our bread rise, and it produces ethanol. Now when we're baking bread, that ethanol gets baked off, it evaporates, and if we're baking beer, it makes it an alcoholic beverage. In our muscle cells, something different happens. In our muscle cells, the electrons actually get passed back to pyruvate, and it makes lactate, which converts to lactic acid. So in our muscle cells, no carbon dioxide is produced by lactic acid fermentation, no ethanol is produced by lactic acid fermentation, but the purpose is still the same. It strips the electrons out of NADH to regenerate NAD+, so that glycolysis can keep going and a little bit of ATP can be made. Now, if we compare fermentation to aerobic cellular respiration, they start the same. They do glycolysis. They make 2 ATPs from glycolysis, but The difference is during fermentation, you just get those 2 ATPs from glycolysis and that's it. Whereas if you do all of cellular respiration, you're going to get 32 ATPs per glucose molecule. So you get way more ATP. So organisms that are able to switch between these two are going to do aerobic cellular respiration when they can, because they'll make more ATP, but can switch to fermentation if there's no oxygen present, because some ATP is better than none at all. So that switch point is the pyruvate dehydrogenase complex. If there is oxygen, aerobic cellular respiration will happen. If there's no oxygen, then pyruvate remains in the cytoplasm and fermentation occurs. So organisms that can switch between aerobic cellular respiration and fermentation are called facultative anaerobes. Some microbes are obligate anaerobes, meaning they can't actually survive in oxygen. They have to do fermentation or anaerobic cellular respiration, and oxygen is actually toxic to them. So this includes like the bacteria that produce cavities. The reason that we brush and floss is to break up those plaques so that oxygen can get in and is toxic to the bacteria there, and they won't form holes in our teeth. Just a few more quick points to wrap up Chapter 9. So we started with our nice, neat, balanced chemical equation where glucose is always our starting molecule. But of course, proteins, other carbohydrates, and fats also enter cellular respiration at different points. Okay? If we think about the amount of energy that's available in these different food sources, a gram of carbohydrates has the same amount of energy as a gram of protein. We can get the same amount of ATP out. But the same amount of fat by mass, okay, produces twice as much ATP. So we get twice as much ATP per gram of fat. Cellular respiration is regulated by a negative feedback system like many processes in biology. So if there's lots of ATP, ATP itself actually goes back and inhibits one of the first enzymes in glycolysis called phosphofructokinase. And so it stops burning the glucose, and that extra glucose can get converted into glycogen and stored as carbohydrates in our muscle cells, or converted into fat and stored for later. On the other hand, if we use up all our ATP, it gets broken down into ADP, and if it gets broken down further into AMP, AMP actually positively regulates phosphofructokinase and speeds up the first steps of cellular respiration, so more ATP can be made. Respiration is so important to many, many aspects of biology that we should be able to think about how this transfer of energy can be used by organisms in different ways. So one example of this is brown fat in newborn baby mammals. Okay? What happens is they burn fat and they release carbon dioxide, but they don't make ATP with it. Okay? So what happens if you are making carbon dioxide without capturing the energy of the transfer of electrons to make ATP? So stop and think about that for a minute. Well, what happens to the energy? If it's not being captured, then it's being released. And so the function of this structure is to just generate heat to keep naked baby mammals that haven't grown their fur yet warm. Okay? So the process of oxidation reduction is still happening. The electrons are still transferred. The energy is released. And the function is to release that energy as a form of heat. Lots of drugs and toxins target cellular respiration because without cellular respiration, you're going to die really fast. One example where this is utilized is diet drugs like dinitrophenol. The way that dinitrophenol works is it's an uncoupler. It facilitates diffusion of protons. So what that means is it makes the membranes of the mitochondria leaky or permeable to hydrogen ions. So instead of going through ATP synthase to make lots of ATP, the hydrogen ions can leak out in other directions. Okay? So what happens in this case is, one, energy is released as heat, okay? And two, you make ATP, you make less ATP, okay? So this works as a diet drug because you can eat lots of food and you burn it and you release the energy without capturing it to make ATP. Of course, you can overdose on this. It's the same as arsenic poisoning or cyanide poisoning, and the fact that if you take too much, your cells won't be able to make enough ATP to survive. So in this question, it says if glycolysis and the citric acid cycle are staying the same, what will happen? Well, you will make less ATP, but you'll still be consuming oxygen at the same rate if those other processes are still happening. And your body will try, so if, as in the question, glycolysis and citric acid cycle are staying the same, ATP synthesis will decrease and oxygen consumption will be the same. However, in reality, your cell, what's going to happen is you're going to start breathing more to get more oxygen to try to keep up ATP production in order to make enough ATP to survive. More thought problem for you. If you did alcohol fermentation instead of lactic acid fermentation, which of these is a correct description of what might occur? So go ahead and read through the options, pause the video, and then we'll talk about the answer. All right, so if you're doing ethanol fermentation versus lactic acid fermentation, you still make the same amount of ATP, okay? You would still recycle NADH to NAD plus at the same amount. You would release more carbon dioxide, not less, and you would also produce ethanol, which means that you might get drunk while you were exercising. So the right answer is actually C. However, there's no mutation that's going to spontaneously occur in you that's going to allow you to start doing fermentation. We are evolutionarily far separated from organisms that can do fermentation, so this mutation's not likely to show up anytime soon.